Hi, I'm Hannes Stärk, an MIT PhD student working with Regina Basilei and Tom Jakula, and I'm excited to be here today on the behalf of this team and to be presenting BoltzGen, our new binder design tool. And to motivate the need for such a model, I want to start off with an example. What we see here in blue is the third telomerase protein, a protein that our collaborators have identified as an important target against glioblastoma. And in these glioblastoma cancer cells, what this protein does is to repair DNA. And now what we want to do together with our collaborators is to design a peptide like this, the yellow peptide here that binds at the specific site that displaces the DNA such that the protein can no longer repair the DNA for the glioblastoma cells and the cancer cells no longer survive. But now, for just this one example, the existing frontier models would already fall short. First, they do not support the required constraints that the human experts here in this collaboration want to impose on the designed peptides. And second, third telomerase is a novel target in the sense that there are no existing proteins or no existing peptides that are bound to this, no structures of that are bound to the third telomerase protein yet. And the existing frontier models, they have all been usually evaluated in the scenario where the binder or the targets that we have at test time is already in the training data with existing bound structures. So what we now want to do with BoltzGen is to expand the designable space of binder design along two axes, with the first one being generalization across modalities and constraints. We want to have one model that can design nanobodies, peptides, proteins, and target proteins, small molecules, DNA, and support all the required constraints that our collaborators might want to impose on the designs. And then on the second axis, maybe even more importantly, we want to be able to generalize to what I already called novel targets. And what I mean with novel targets are targets that are not in the training data with existing bound structures. And this is very much unlike the evaluations that we have seen for the existing frontier models, where the targets that we evaluate on are usually in the training data with existing bound structures. And what we think is necessary toward that goal is to, one, get closer toward emulating physics, and two, have a model that is strong at reasoning about structure. And now, before I lay out the model that we developed toward those goals, I want to take away that to test this, whether we achieve these generalizations along these two axes, we have to bring our designs, the outputs of these models, the binders, into the real world, into physical reality, and test them in the wet lab. And we do this in collaboration with by now more than 26 wet lab collaborators, of which 8 campaigns have already finished. And this gives us results across 26 targets, which I'm happy that I can show them to you later. But now back to the model. So let's remind, what do we want to achieve? One, we want strong structural reasoning. Two, we want to get closer toward emulating physics with our model. These two goals were toward the end of getting closer and being able to generalize to novel targets. And then three, we want full controllability. So we want to be able to control the design, the constraints on it as desired by our human experts using the program. All right, then I will now dive into point number one and just make this very explicit. Why do we want strong structural reasoning? Well, if we're doing design, then this task means that we have a target A, and now we want to design the binder B, C, D and choose out of these infinitely many possibilities what the binder could be. And choosing out of these possibilities means discriminating between them, right? We need to discriminate between B, C, or D, and so on, whether these are the most favorable binders. And discriminating requires understanding how A would interact with B or with C or with D and so on. And this understanding how proteins interact, this is exactly what the folding task is, the structure prediction task. And maybe in some other words, we can say that if we want to do strong structure-based design, then we need strong structure reasoning. I think that's reasonable. And now point number two was that we want to get closer toward emulating physics. And what I mean with that is that even if we wanted to only design nanobodies to bind to proteins, then also in this scenario, we would want to train on protein-protein interactions, for example. because our models learn to emulate physics based on example, and thus we want to provide our models with as many examples as possible where these physical patterns still hold. And this will be the case across modalities and across contexts. And also, if we apply augmentations to this context, then the patterns that still hold in all of them, patterns that are always true, are, for example, physical interactions. Okay, point number 3, just explain based on an example, what do I mean with controllability? Here we have, for example, that our collaborators want to design this dark red CDR3 region while keeping the rest of the scaffold fixed and sampling the CDR1 and 2 from a fixed library. Sometimes they want to design all CDR regions of this nanobody and keep the structure of the yellow part and the sequence of the yellow part fixed. Sometimes they want to redesign everything. And now this is just one piece or little pieces of the spectrum of possibilities along one control option. And these spectra, so to say, also exist along many other control options. All right, now we have these 3 points. How do we achieve the 3 points? Towards strong structure reasoning, we unify the structure prediction and design tasks. Toward emulating physics, we come up with a training distribution that tries to exercise the model on as many contexts and modalities as possible on the available training data. And toward fault controllability, we have a design specification language that is very expressive and allows the user to essentially specify anything they might want to desire. Okay, but then I want to take away that this model that I'm talking about now is only the beginning of the larger BoltzGen pipeline. The first step, the maybe most important step, but the others are also important. So how does this first step now work? What we're seeing here is the denoising trajectory, the diffusion trajectory to generate one sample, one binder. And what is happening here is that we're simultaneously folding the target. We're reasoning about how the target changes its conformation while binding the binder that is simultaneously being designed. And we're generating the binder, which means we're simultaneously producing its structure and its sequence. So we have a co-generation problem going on here, where we're simultaneously producing a continuous structure and a discrete sequence. And the way this is realized in our model is that we treat the designed protein and the target protein identically, where for both of them, we have only noise as input to the model. And it also only predicts 3D structure. The sequence is just an additional conditioning input to the model. And this is quite different from how frontier models have done this in the past, where usually the structure of the or the target protein and the designed protein would be treated quite differently, where for the target protein, the denoiser, the diffusion model, always already takes the clean structure as input, and then it doesn't perform any folding, right? It just copies over the clean structure, which means the model does not learn these nice features that we want to learn from folding, this strong structure reasoning, and having these expressive features that capture how residues will interact with each other. So in our architecture that is arguably quite a bit simpler and more scalable, we are completely folding the target and the design from scratch. And we are also really only doing folding, only producing a 3D structure for the design, not a sequence. So this comes with the nice benefit that 1, we can fold the target. We can also predict how the target changes its conformation. Very nice if we, for example, want to bind to disordered regions. And 3, we're still doing this joint design of simultaneously producing the structure and the sequence, because in the structure that we're producing here, the sequence is embedded, and this discrete diffusion, this discrete generation process is embedded in this continuous, or in the same continuous space that is used for the structure. And the way this works, I will quickly explain based on this diagram here. Because for us, a designed residue, now, for example, a designed isoleucine, it doesn't have the usual 8 atoms that are in an isoleucine, but it has 14 atoms. Every designed residue has 14 atoms. It is just that now the isoleucine has an additional 6 virtual atoms that are placed at the same coordinate as the oxygen of the isoleucine. This means that at inference, the model starts out with 14 randomly placed coordinates. It denoises them to end up with this point cloud, where we have 6 or we look at the oxygen and see that there are 6 additional atoms very close by to it. And we look into our table and realize this must be an isoleucine. So we have sort of turned the design task into a structure prediction task, right? Because now we're only predicting generating a structure, and this structure contains, encodes also the discrete residue types. Now to point number 2. We have this training task distribution across modalities. And here I'll just remind, right, we said we now want to train on all these modalities because the patterns, like physics, they generalize across them. And how this then looks like would be that we draw a structure from PDB. We randomly select a center residue to crop around. Then we obtain this crop 3D structure. And then on this cropped 3D structure, we, for example, with a certain probability, only train with the folding task. The model gets the sequences as input, has to output the 3D structure. There's no design going on. With a certain other probability, we perform the design task, where now a subset of the residues, a random subset, it could also be the other chain, it could be completely different residues, is in yellow now selected to be the designed residues, which means the model does not get a sequence as input. And now it is tasked to produce this 14 atom representation where every single designed residue has 14 atoms. And sometimes with a certain probability, we also feed in the structure of the target. Sometimes we feed in the binding site specification. All these are sampled from a distribution, and all the other conditioning possibilities and contexts that we have are also sampled to exercise the model in as many contexts on this data as possible. And this distribution also changes over the course of training. Like, for example, at the beginning of training, we only train with structure prediction. Okay, and now to point number 3. What do we want to control? And here I'll just step through examples again. So, for example, we want to be able to do this partial design where we say, this should be a cysteine, this should be a cysteine, and only the other residues should be designed. Or we want to say it should be a helix. We want to say there should be covalent bonds between these cysteines here, for example, and another small molecule, or you can specify any other type of covalent bond. We do not just want to be able to say what is a binding site of the design that we're generating, but also what is a not binding site, in the sense of this is a residue that the binder that we produce should be far away from. And then lastly, based on an example, I will lay out how our structure group conditioning or our visibility control here works. So these structure groups or visibilities, they are now explained based on this example where we want to design a nanobody, orange and yellow, that should bind to the target in blue, these 2 chains. And then, because we want the nanobody to be free to move with respect to the target, but we want to still specify the structure of the scaffold of the nanobody, we have this concept of structure groups, where you can say that everything that's in the same structure group, so like structure group 1, sees each other and has its structure with respect to each other specified, but everything that's in a different structure group is not or is free to move with respect to this first structure group. And everything in structure group 0 does not have a structure specified. And this is all put together into like a design language in YAML files that you can use. But now I mentioned that this is only the first part of the pipeline, right? So what we do in practice is, with this diffusion model, we generate 60,000 designs. Then we produce an inverse folding, or we run an inverse folding model on top of it. And happy to discuss that in detail if you want to hear more about that. And then we run this refolding step where we start with the designed structure and then extract the sequence from it, refold it with a structure prediction model, and then look at, does the refolding model agree with the design structure? And we want that to agree. We have an affinity prediction step that I'm skipping because it's just another learned model. And then we have this analyze step here, where we count the number of salt bridges, hydrogen bonds, look at the interaction surface area, and we look at a whole bunch of other physical metrics. And all this is then combined in the filter step, where we rank, or we combine all the metrics to obtain a final ranking and run a diversity quality optimization algorithm to select a final diverse set of high ranking designs. And here is an example of what this denoising diffusion trajectory looks like. But now I will get to results, and the first set of results that I'll show are structure prediction results. So there's no design going on, right? And in this structure prediction results, we're now comparing BoltzGen with a state-of-the-art structure prediction model and see that our design model can also do structure prediction and can do it at the state-of-the-art. And this holds across modalities. But now to the mentioned real-world wet lab validation, where we have all these collaborators, 8 wet lab campaigns across 26 targets have finished. And to begin with, I want to show the results on what we call novel targets, where we select 9 targets that have less than 30% sequence similarity to any bound protein in PDB. And then for each of these targets, we design 15, or we test 15 designs, and then check how many of them express, how many of them are binders, and what is the best binding affinity. And we find that for 9 of these novel targets, we get successful binders in the nanomolar range when designing nanobodies. And there's also no off-target binding against human serum albumin as a check that we really only bind to the targets that we want to bind to and not the sticky off-target. The same holds for proteins. But now here an interesting case where our collaborators had this protein with in blue a structured region and in green an intrinsically disordered region. So we designed a peptide that should bind this unstructured region. And the model thinks that with the peptide in yellow that it designed here, that then the green region will fold up into the structured region like this. And we test 5 designs, and it turns out that one of the 5 indeed actually works. And here in these experiments, we test, can we design binders to neutralize the activity of 3 antimicrobial or hemolytic peptides. And again, when just designing or testing 6 peptide or 6 designs in the wet lab, we obtain nanomolar binders against 2 of them. And for each of the targets, we also obtain designs that neutralize the bioactivity for the target. Okay, with this, I conclude, right? We set out to expand this designable space, and to do so, we turned the design task into a folding task. We exercise our model on as many contexts on the training data as possible, and we have this design specification language. And then we now tested and experimentally validated our model in 8 wet lab campaigns across 26 targets, and I only showed a few of these results, but more are available in the paper. With that, we now maybe have a model that is good at designing high affinity binders, but I want to take away that really importantly, this is only the first step toward generating a therapeutic. For a therapeutic, you also would need to deal with immunogenicity, delivery, your target would have to not be degraded if you ingest it, you would need to be selective, and these are of course also things that we are working on. These developability properties or generating designs that have nice selectivity profiles, and also other things like other modalities where we, for instance, want to be able to design RNA to bind to proteins. Okay, and then lastly, I want to remind that I'm only a very small part of this larger team that did this work, and especially a big thanks to our wet lab collaborators that took a chance on our model and tested its designs. With that, here are the links to our resources, and I'm happy to answer any questions if you reach out to me.